welcome to State of the Science. This is a new way of talking about research in the field of type 1 diabetes. This series is really a celebration of the power of debate and discussion that women scientists in type 1 diabetes research bring to the table. We have a global audience of scientists here today, and we thank everyone for joining. I'm really going to briefly introduce the panelists. The panelists are going to sort of set the stage for the discussion, and then we're going to weigh in, ask them to weigh in on their thoughts on sort of like 3 to 4 pressing questions in the area. And it's going to be a sort of a free-flowing cross-idea exchange, town hall, respectful debate. And we're going to pause at 2 different times to sort of like send out polls to the audience and have them weigh in on what we're talking about today. So if you have a couple of pro tips for attendees, if you have a question, put it in the Q&A, or you can submit questions throughout the presentations, and participants can upvote favorite questions. And at the end of the session, we'll answer questions as time allows. So this event's being recorded live, and it will be housed thereafter for further viewing. So in the interest of saving most of our time for discussion, I'm going to do very brief introductions of each of our scientists. We could actually take up like the whole time discussing each of them and their work. They're super valued and integral members in the T1D research field. You can connect with them directly on our website via the collab, where the majority are members, or you can reach out directly. So I'm going to start with Dr. Bridget Wagner. She is the director of pancreatic cell biology and metabolic disease in the chemical biology and therapeutic sciences program at the Broad Institute of MIT and Harvard, where she's also an institute scientist. And her group's research focuses on chemical biology of diabetes with the aim of identifying small molecules capable of increasing pancreatic beta cell number and function, and the ultimate goal of discovering a new therapeutic approaches for diabetes. So welcome, Bridget, Dr. Wagner. And then Dr. Kendra Venhik, she is coming to us from University of South Florida. She's a professor of epidemiology at University of South Florida. And she is an epidemiologist and applied biostatistician. Her main focus is on the early life prevention of islet autoimmunity and type 1 diabetes. She's a collaborating investigator in many type 1 diabetes studies, including SEARCH, TEDDY, TrialNet, DPT1, and Discover studies. She also serves as the co-chair of TEDDY Publications and Presentation Committee, and oversees dissemination of scientific content for the TEDDY study. Big data there. Dr. Milena Pavlovic, she is coming to us from Oslo. Actually, she's a graduate student. I'm sorry, I misspoke. She's on her way to becoming a doctor. She is, together with her team, she's developed ImmuneML, which is an open learning system for ML analysis of adaptive immune receptor repertoires. And this novel system has multiple applications to type 1 diabetes research. Dr. Melanie Shapiro, she's coming to us from University of Florida Diabetes Center. And she is a postdoc at UFDI and a recent winner of the D Challenge, which was a data-based hypothesis creation challenge. Her current works focus on the use of precision medicine genotyping microarray data to investigate the role of T1D risk loci in promoting immune and pancreatic phenotypes associated with type 1 diabetes pathogenesis. Dr. Maryann Martone comes to us from the NIDDK, also from dkNET, and she brings a deep understanding and a long career in neuroscience and data to this conversation. She leads the NIDDK Information Network, which is called dkNET, which is a portal for connecting researchers in digestive, kidney, and metabolic disease to data, tools, and materials. And last but not least, it's a little bit, I'm having a little bit of fuzziness here, but hopefully that'll dissipate. So Dr. Alma-Martina, she's MD, PhD from Stanford. She's an instructor in the pediatric division of stem cell biology and regenerative medicine at Stanford, where she's working to uncover molecular mechanisms governing type 1 regulatory T cells, or TR1, differentiation and function, and use this knowledge to design a TR1 cell-based therapies for hematopoietic stem cell transplantation, cancer immunotherapy, and autoimmunity. Distinguished panelists, thank you so much for joining us. And I'd like to ask if you could set the stage for this discussion today, which is cutting edge technologies and in silico solutions for the T1D Research Laboratory. So I'm going to, let's see if we can just start right at the top. What are the most powerful tools in the toolbox right now regarding T1D research? You know, and you can even kind of comment on what are the most difficult to master. So if Dr. Wagner could sort of weigh in.
Sure. So I wanted to talk a little bit today about cell-based and therapeutic-based technologies, of course. And I think that, so I'm involved in the Human Islet Research Network, or HERN, which is funded through NIDDK. And there are just so many, it's a very exciting time. There's so many new technologies that are being developed that are going to, I think, have an impact on type 1 diabetes, both from a modeling and understanding standpoint, as well as a treatment standpoint. And in particular, I'll highlight two areas of interest. One is developing biomimetic models of the human islet, and this involves a lot of microfluidics, co-culture of islets with the immune cells from humans as well, and then using interesting high throughput, high resolution readouts of different perturbations. This allows us to test many different conditions and many different potential treatments, but with the idea that in particular it's using human tissue, and I think that's going to be very, very important moving forward as we think about therapeutics, just with the difference between a lot of mouse modeling versus using human sources of cells. And in particular, and I know I'm going to forget people, it's not meant to be a comprehensive review, but at the University of Florida, there's actually quite a great group of folks that are working on these biomimetics, islets on a chip idea, in particular Sherry Stabler and others. The second big area I think that's worth highlighting is targeting. So if you have developed a therapeutic, and for example, we are developing small molecules that can promote beta cells to regenerate and proliferate, but what's really important is making sure those potential drug candidates get to the right tissue. Many of the phenotypes we're promoting, would be terrible to have happening in the whole body. And so figuring out ways that we can deliver either small molecules, macromolecules, or genes even directly to either the islets or the immune system would be really, really valuable. And there's a lot of work going on in AAVs, in particular, looking for capsids that can home to the beta cell, for example, and as well as work in aptamers. So using RNA aptamers to see if we can deliver and conjugate and deliver, for example, siRNAs or other types of therapeutic reagents. And some of the work I've been doing in collaboration at the Broad has been in the prodrug area. So the idea that you would have a compound that is inactive and only unmasked and revealed as the active ingredient, for example, in the beta cell. All of these are a long way off, and they're really lab work. But I think they show a lot of promise in terms of new technologies. So I'll stop there, but we can talk more about those as we go on. Thanks.
Absolutely. There's a lot there to unpack. How about Dr. Venhik? I know you have a slide.
Yeah, I have a slide. I'll show it in a second, but I want to start off with, and I'm going to come from a more epidemiologic viewpoint in terms of some of the techniques that I see out there. And one of it is really our profiling technique. I think it's really come a long way. We've made a lot of advances in profiling, specifically because we've been able to obtain large-scale measurements. And I mean that from both the omics, so biomarkers, as well as from the environmental exposures in both children and adults in very different geographical areas, especially in large populations. And this has really helped us to begin to develop that, the profiling or developing signatures across the board, and it's allowing us to power up our studies so we can develop more predictive models. And one in particular would be the refinement of our genetic risk scores. And I think that's really taken us a long way because we've been able to not only identify common features among population that kind of weight down or attenuate our findings, but also see those specific variants, so specific genes that are very specific to a subgroup. And then within those subgroups and reducing heterogeneity, which allows us to look at a more homogeneous risk group. And that's really getting more closely to being able to look exposures and omics data that will allow us to get at the disease progression pathways. So, and just to give one quick example, I'll share my slide. Uh, can everybody see that slide? Yeah, this was a study. So in terms of like really powerful tools, and this goes along with what Bridget talking about cell type of work. A group at Teddy worked on this, and this was really an interesting study that used single cell RNA-seq. And really what it was able to do is help us understand different cell proportions and gene expression across all levels within a specific cell type. And what I found really interesting about this work and what it's really helped us move forward is that you can see, and very much in these radar charts at the very top, you can see that there's a spike here in the children who presented with IA first, so the islet autoimmunity antibody first versus the GATA first autoimmunity. You can see definitely a different signature. And those signatures related to two specific cell types that were similar across both groups that progressed to type 1 diabetes in NK and CD4 memory T cells. But what we really did see is a pattern that was different based on those first appearing antibodies, where when we looked at it as a full cohort and not looking at it by the subgroup of islet autoimmunity, we did not see a difference. So this new technology, I think, is really helping us peel apart what's going on and how to subgroup and reduce variability that will help us to really start to look at this complex disease.
It's fantastic. Okay, that's great. And I guess if Dr. Martone wants to talk just for a minute and share, like, what is your, what are the most powerful tools in the toolbox for you regarding T1D research right now?
Thanks, Monica. I just wanted to have one clarification. I'm not with the NIDDK. I'm at UCSD, but I do run the, along with Jeff Greta, the NIDDK Information Network. So our focus and specialty is connecting researchers to resources, and those resources include reagents, organisms, cell lines, but data and tools. And so if you ask me what the big opportunities are now, it's really the area of being able to bring these powerful new technologies, artificial intelligence, machine learning, deep learning, to the biomedicine in general, but type 1 diabetes. These types of techniques are going to let us look across massive amounts of data, looking for patterns, looking for predictiveness. It is beyond the capacity of individual researchers to be able to do this, so we need to be able to bring different data sets that are produced by individuals together so that they can be analyzed together. I think that this type of in silico biology, which just at its infancies, and it takes a while to be able to develop the expertise to be able to avoid pitfalls, biases, other types of things that we're going to get. But what's exciting about it, in my view, is building this on a pool of publicly available data that allows us to take the entire intellectual capacity of the world, the really smart data scientists that are all over the place, and bring them to this question, which is very personal for many people, right? Many people have relatives who are affected by type 1 diabetes. They are affected by it themselves. And you see this motivated workforce out there. So dkNET itself is really devoted towards making sure that people as an information network understand the resources that are out there. We participated in the D challenge that Monica mentioned to really encourage people to dive into these online resources that are there. But we're also devoted towards making these resources better, right? Letting scientists collect better data, understanding what it means to share data effectively. There's something called the FAIR data principles, that you want to make data findable, accessible, interoperable, and reusable. And dkNET is leading the way there in the area of diabetes to say, if we're going to produce these resources, these data resources, put them out there, make them available, be able to combine them with other data, then we need to pay a lot of attention of how we collect data in the laboratory and make it available. So I think that these are where the exciting opportunities are. They're going to let us look not just, for example, at type 1 diabetes versus control, but type 1 diabetes versus everything, right? Because it can ingest enormous amounts of data. And I think that that's going to lead us to new insights. But it'll take time, but more importantly, it will take data.
Yeah, well said. And we, recently partnered with the, dkNET group to, try out this whole idea of the D challenge, where we had several teams diving into data to try to develop some new hypotheses. And that was a really great experience. Very exciting times at the dkNET. So thank you again for sharing all that with us. How about Dr. Sepika coming from Stanford?
Hi, yes. My passion are antigen-specific regulatory T cells and the potential of these cells that we can use them as a cell therapy to really reprogram the immune system from the state of intolerance, such as in autoimmunity and diabetes, to the state of tolerance. And I'm currently studying these cells in the context of allogeneic hematopoietic stem cell transplantation, but the findings from that field can be easily translated to autoimmune diseases. So I have a few slides just to show the advantage and the power of studying antigen-specific T cells by means of TCR sequencing. Do you see my screen?
Yes, perfect.
Okay, great. So the T cells that recognize and then destroy the insulin-producing islet cells, they are really the key, play a central role in the pathogenesis of type 1 diabetes. But the regulatory T cells that recognize the islet autoantigens are protective, and we can transform them into cell therapy that can be applied early on and then stop the progression of the disease. The antigen specificity of each T cell is determined by their T cell receptor, or the TCR, which interacts with the target peptide HLA molecule complex. And these T cell receptors are incredibly diverse. And each T cell has a unique combination of TCR alpha and beta chains. And this combination, which we call the clonotype, is passed on to the cell progeny as T cells divide and can serve as a natural molecular barcode that can be used to identify T cells with the same antigen specificity. And this can be used for immune monitoring and also for some, for example, for identifying and predicting autoantigens. So this TCR sequencing, it can be done either from genomic DNA, which is useful when the starting material is not in optimal conditions, and also a TCR-seq from genomic DNA is very quantitative because T cells have only one copy of each gene. On the other hand, it can be also done from RNA, which can be more sensitive, as each T cell has multiple copies of the TCR alpha and beta chain mRNA. So in these type of studies, it's important to differentiate between pathogenic clonotypes of T cells in type 1 diabetes, and also our autoreactive clonotypes on T cells that can sometimes be found in healthy individuals, which means that these TCR sequencing studies should be combined with functional assays to confirm the autoreactivity and pathogenicity of these clonotypes. And so here are just two papers out of many that as an example of studies of antigen-specific T cells in a mouse model of diabetes in NOD diabetic mice and in T cells from human pancreas with patients with type 1 diabetes.
Yeah, it's great to see that there's a lot of collaborative effort involved in those papers all the way across, Vanderbilt, Florida, Stanford, City Pope. It's really nice to see that type of effort. And then I guess I'd like to go to Dr. Shapiro.
Great, I'll pull up my slides. So first, I want to thank Monica for inviting me to speak today about my perspective on bioinformatics and type 1 diabetes. This is a really nice opportunity. So thank you. And
thank you for taking your time out of the laboratory work, because I see you're in the lab right now. So.
I'm in the office. That's why I'm wearing. It's a shared office, so that's why I'm wearing a mask.
Right.
All right, so you should be able to see my slides now.
Great.
All right, so I personally think that the most powerful bioinformatics tool that we currently have available for type 1 diabetes research is the ability to perform quantitative trait loci analysis. So QTL analysis answers the question of whether variants, specifically in our case, type 1 diabetes risk variants, correlate with modified gene expression or function. And what's important to note is that variants can have context-dependent effects. So certain SNPs can only have, impacts on gene expression or function, maybe in a particular cell type or in the presence of certain environmental stimuli. So you can kind of imagine how there are so many different kinds of QTL analysis experiments that could be designed to answer all these different questions about how type 1 diabetes develops. And really the power here is that these are correlative studies that can identify specific associations between genotypes and phenotypes that contribute to type 1 diabetes, whether that means certain immune phenotypes or phenotypes, let's say in the pancreas. And then we can take those and actually test them in the lab. So we can use these to design wet lab experiments to actually demonstrate the causal effect of that variant in actually causing that phenotype. And so what I wanted to just briefly cover over the next 2 slides is a case example of work from 2 members of my lab, actually another postdoc and a staff scientist, where one of the members found a correlation between a genotype and a phenotype, and then another member performed the causal experiment to show that that variant actually did cause that immune phenotype. So the genotype and phenotype in question, the genotype is the risk allele that is tagged to the PTPN22 gene, the SNP being RS2476601. And a lot of the people on this call are probably very familiar with this, since it has a really, really high odds ratio for type 1 diabetes. So PTPN22 is a phosphatase that regulates T cell receptor signaling by dephosphorylating several substrates in the TCR signaling pathway. As you can see here, it dephosphorylates LCK, ZAP70, as well as the zeta chain of the TCR. And this particular SNP that's associated with type 1 diabetes is actually a missense mutation that causes a change from an arginine to a tryptophan in the C-terminal domain of PTPN22. And this particular domain is really important for PTPN22 function because it's responsible for protein-protein interactions between PTPN22 and this protein known as CSK. And so this protein-protein interaction normally occurs in resting T cells to sequester PTPN22 to basically make it localized near the TCR and perform its function of inhibiting TCR signaling. So in the case of the risk allele, you lose this protein-protein interaction, and then PTPN22 can kind of just float away, and you get this unregulated TCR signaling. At least that's the hypothesis for how this SNP could induce loss of function. It's a little bit complicated because the field is in a little bit of disagreement whether the SNP actually causes loss of function or gain of function. But the data that I'll be showing supports the loss of function hypothesis. So starting on the left, we're looking at, I'm sorry, this is a little bit blurry, but we're looking at some correlative data. So this is from Robert Sharp, another postdoc in the Brusco lab. And this figure is showing proliferation data from PHA stimulated T cells that were isolated from either healthy subjects or subjects with rheumatoid arthritis. And they either have the protective allele at the SNP, which is the G, or the risk allele, which is A. And the minor allele frequency for this SNP is actually pretty low. So you can see that there actually aren't any subjects that are homozygous for the risk allele, but we do we do see subjects that are heterozygous. So there is this comparison of heterozygous to the homozygous protective. And what you can see is that in the rheumatoid arthritis affected individuals, that the subjects with the heterozygous genotype actually showed this enhanced proliferation of their T cells as compared to those that were homozygous protective. And so this is maybe a little bit of a stretch because this, as an example, because this actually wasn't performed as a QTL analysis, but you can imagine how this kind of study could be performed as a type of QTL analysis. You could have looked at all of the type 1 diabetes risk SNPs and found that PTPN22 was the one that affected T cell proliferation levels. So here on the right is data from a staff scientist in our group, Dr. Dan Perry. And what he actually did was he induced lentiviral overexpression of PTPN22, either protective or risk variants. So the R being protective and the W being risk. And this is all in individuals who were healthy subjects who endogenously expressed the protective allele. And what you can see is that these data actually recapitulate what was observed in the correlation study by Dr. Sharp. So the overexpression of the risk allele actually led to increased proliferation of T cells compared to overexpression of the protective allele. So what I'm hoping is that using this case example that I've convinced everyone about the power of QTL analysis for identifying genotype to phenotype associations that are worth validating in wet lab experiments.
Yeah, that's fantastic. I love how we've done some high level discussion of these different tools, and then you got very granular here to really show the case for using this type of tool. So that's really great. Thank you very much.
Yeah.
And then how about let's hear from Milena Pavlovic from University of Oslo regarding what is ImmuneML and why is this a powerful tool in the toolbox for T1D research right now?
Thank you first for having me here, and I will just share one slide to briefly illustrate this. So ImmuneML is a software platform for machine learning analysis of adaptive immune receptors in repertoires, which sounds like a lot, but it actually focuses on two main use cases. So in the first use case is just looking into the receptor, the adaptive immune receptor itself, in combination with VD and the J gene, and trying to come up with machine learning models that are able to predict the specificity of that receptor. And of course, for that, we need a lot of data. So ImmuneML is also integrated with some of the public databases for this purpose. And the aim is to, based on that, on experimental but also simulated data, be able to develop machine learning methodology that can predict the specificity and also find the patterns in the receptor itself to describe what is it that determines that specificity. With, of course, then, if we have that type of information, we can also look a bit further into, for example, generation of antibodies with the given specificity. And the second main use case that ImmuneML covers is predicting the immune state based on adaptive immune receptor repertoires. So based on between 100,000 and a million receptors that we can obtain from either bulk or single cell sequencing, the idea is to develop methods that look into these repertoires and find the patterns that distinguish the diseased individuals from healthy ones, and to find what is it that determines their immune state. Of course, this is a challenging problem. There is no need to convince anyone of that, but it is also a very challenging problem from machine learning side, and which also makes it very interesting for us computational researchers. And so there are already some approaches that do this. Some are based on different deep learning techniques, and for example, DeepRC that has been developed at the University of Linz. And it is available within ImmuneML platform, so it can just be trained on the new data. And many other methods are currently being developed to show that something like this is possible in principle. Of course, with the aim that we can do some sort of broad testing based on the developed models. Yeah, and one thing I would like to emphasize about ImmuneML is that it is open source and it is freely available to researchers to use and also extend. And we have a web server as well that is adapted more to also immunologist users to try it out before getting really deep into the details of how specific machine learning models work.
That's great. Excellent. And it's great how it goes, you know, kind of hand in hand with what Dr. Martone was talking about earlier, the concept of open science and how important it is to moving science forward. At this time, I think I would like to, I appreciate everyone's perspective and, you know, kind of walking us through the most powerful tools in the toolbox, in your opinion, regarding T1D research. Obviously, they're all very important. I'm going to send a quick poll to our audience right now. Oops, maybe I'm not. Oh, all right. Well, let's see if we can, maybe why don't we have a quick discussion about, you know, what we've said, I guess, so far. And I'll try to get this poll going. I don't know what happened to it. But yeah, so I mean, in terms of like, you know, what applications will, the poll was really like, what application will bioinformatics tools most effectively contribute towards? And we were offering these answers: biomarker discovery efforts, disease endotyping, identification of precision medicine therapeutics, and interdisciplinary researchers mining T1D specific data. I mean, what are your thoughts on this kind of, you know, series of questions? Let me see if I can add it to our chat. I'll add it to everyone. You can see it in the chat. Everyone's able to see it there. Does anyone want to kind of comment on that? I'll try to resurrect our poll. Don't all jump at once.
I mean, I think in theory it would be all of them because I think that bioinformatics, data science is able to, all of these require looking at large amounts of data and looking at, you know, making predictions. And I think that that's what these methods are for. So I think that they can impact all of them.
Yeah. Does anyone else have a you know, kind of input on that?
I would agree, and maybe add to this, you know, public health efforts that are focused on preventative medicine. Because, you know, type 1 diabetes, when the destruction starts, it is something that is hard to reverse. And hopefully we will be able to do that with recognizing patients early on in the disease. But it would be ideal that there is an easy to use and cheap and applicable method that we can screen children who might be at risk of developing type 1 diabetes before the disease even starts.
Okay, now I'll launch this to the audience. This is a anonymous poll also, so you can sort of, you know, answer it on your under your anonymous banner. Can everyone see this?
Yes.
Okay. let's see what people have to say. It looks like at least people are starting to answer now. Looks like biomarker discovery event efforts is in the lead, and that's kind of interesting. Here we go. Let's at least get 50% of people. Once 50% have answered, and then we can comment a little bit on this whole idea of biomarker discovery efforts and how we can, how that might get enriched and extended, I guess, into the community. I mean, obviously it's happening already. Here we go. I'm going to publish this. So here's the poll, and you can see that, you know, it kind of goes A down through D. Biomarker discovery efforts are sort of the most popular that people think will really move T1D research forward. I do think, you know, and I think everyone spoke about biomarker discovery efforts because, as you suggested, like that, there's a long prodrome for most type 1 diabetics. And so it would be very interesting to be able to identify who is, you know, who's going to progress, who's not going to progress, how fast they're going to progress. I mean, we did touch on this at the last State of the Science when we were discussing endotypes and whether or not how that can be translated into the market. And I think here's a question, here's a comment from Leandro Balzano. It's prevention is very important. Early detection of biomarkers is the key. Artificial intelligence to determine trajectories would be interesting. So thanks for that. And then I just would, let's see if we shut this.
Maybe if I could ask a question, Monica?
Yeah.
Sure. I have a question for, well, first for Melanie, but really kind of the whole panel. Very interesting about, you know, going from QTL to knowledge about, you know, what a variant effect really has physiologically. And this is something we've struggled with as well. That takes a lot of effort. We're going kind of back to the one gene variant, one student type of process. What are your thoughts and perspectives on how we could make that more streamlined, more automated, if at all, or is this something that we're just going to have to bite the bullet on?
That's an excellent question.
I totally agree. That's a really, really tough question to answer, actually. I think maybe what we need to do as a field is probably, there needs to be more interdisciplinary work between labs to maybe facilitate answering these kinds of questions, because on the one hand, QTL analysis is going to require a very specific set of skills, but then doing these bench experiments also requires a very specific set of skills. And the big problem, I think, is that there aren't a lot of labs that do both, that have a lot of expertise in both.
Totally agree, yeah.
So it's just about collaborating.
Would the solution be to have a core, like a core institute or core lab, where everybody can send, okay, this is the gene, that a variant I want to be expressed, or this is the TCR receptor that I think it's potentially, you know, recognize an antigen. Can you express it for me? Would that help?
Definitely,
possibly, yeah. And I know there's no answer. I was just wanting to stimulate discussion, and it's something we think about across. I mean, it's really disease agnostic. It's any kind of, we find a variant, it's associated with whatever, right?
Absolutely.
Phenotype. Why? Right? That's a huge question, and it's really tough across any disease. So, yeah.
It's almost as if, you know, you've got the NPOD, the tissue repository, and now as the whole field and many fields are getting into data, it's almost like, wouldn't it be great to have a data repository that's specific to T1D? I mean, does that even exist? Is that something that could be built?
Well, there's certainly a lot of them that have
T1D.
been set up. Yeah, for T1D. I'll just share my screen. These were some that if you look at just in the clinical domains, these are all of the type 1 diabetes research networks. So there are efforts here, but even in areas where we feel that we can easily share data across, like in genomics, there's still very siloed. If you listen to people talking from the Global Alliance for Genomic Health, we think that, for example, data sharing of genomic data has been solved, but it really hasn't. And so that you can, it's been solved in some capacity. But as he said, Richard Hausler, who was giving a talk on it, said, if you really want to easily find, you have a variant in breast cancer and you want to go and look at all the variants that are possibly been available, it's still scattered in multiple places. That makes it very difficult to bring it together. There are, I know Dr. Rob Williams at Tennessee set up a resource, a gene network that really was based on not looking at one strain of mouse or two strains of mice or one strain of rat, but rather these BDX mice, where you have all of these crosses, where you have these interesting panels. I think there are about 80 or so of them, and you generate almost every possible cross, and you can actually look for QTLs online using this. Now, of course, that's in mice. I think there's also a rat panel. But I think that it is possible to set up shared data resources where you can specifically address this type of question, because at least in the animals, you can go right back to their specific genotype, right? You can generate, you can look at what traits tend to correlate with which phenotypes. And then, of course, it's available for experimental manipulation because you can actually get those mice, those crosses available. So I think there are some interesting possibilities there. But whether one has setting up a single gigantic resource for all of type 1 diabetes is very challenging, right? There's many, many different data types in there. Things constantly change. There's a lot of technological flux that goes on, but making it easier so that if you have this question, you can go to the resources that are available and answer it is really something that like a dkNET would be interested in helping to tackle, because we are already starting in a very small basis, very small sample. We have something called the Hypothesis Center, which is designed to really introduce people to bioinformatics tools. But if it's possible for us to help going from one resource to the next, go search for a phenotype here, a set of genes over here, bring them together in a computational environment. I think you see that in ImmuneML too, right? There are these sort of tool platforms that you can set up that make it easier to do certain types of analyses. We just have to figure out how to do it.
It seems as if, Kyle Galton's group at UCSD is setting up the T1D knowledge portal, and Noel Bird has the T2D new knowledge portal at MIT the Broad. But, and those are fantastic and they're growing, but it's like what you said. I think it's like there needs to be a target of data resources for type 1, like all things, right, in these different compartments. Here's a good question from Todd. He said, perhaps the way to prioritize studies relates to whether or not it's actionable. That's really important. Drug or antibody target. Do you guys want to comment on that? Like, should we just go after actionable items, or do we want the knowledge base?
I think one of the challenges is also reproducibility. So we find something in one population, and how do we replicate, and getting access to data sets to do that. So in terms of Todd's question related to, antibodies or drugs or whatever the target is, unless we can replicate it before we start moving that direction, it's going to be difficult to give resources to that. And I think that's the biggest challenge. And I know that JDRF has tried to develop a hub with including studies for type 1 diabetes, but I think the difficulty there has been ontologies, so definitions and data types across. Like you have antibodies, but how do they collect them, and what was the interval of collection? So I think that is what I think I struggle with. And coming from Teddy, which has a really nice big data set that's tight, when I look to replicate stuff that I do, it's hard to find a complementary study that I can replicate in. And I think that's really needed now, because we're not able to, sometimes we find something and then we look at a different population that's more general, not as HLA specific, and we have difficulties. So I agree with finding a way to share and having more access to these large data sets.
Mm-hmm. and the standards that you really are talking about, right, in acquiring them. And so these are things in neuroscience, right, that we've been struggling with, because this field is also like neuroscience. It's multidisciplinary, multiple data types, things that you're trying to bring together. And it's very hard to get scientists to think about standards. They don't like to. They don't like to use them. Research standards are also tough because the field changes all the time. But when you start to have a mechanism whereby people will come together around paradigms or other things, even just documentation, best practices, then it allows the field to operate somewhat independently. But at the end, you're not going like this, right? You can start to piece things together in certain ways. So I think that there are practices and things, and in dkNET, these are the types of things we're interested in. But what we haven't seen in this field, like we have in neuroscience and other places, are the convening bodies who get people together to say, look, we have to actually solve this problem at a data level, right? At a data level, we have to solve this problem in order to be able to bring together different labs who we're all resource limited, right? Nobody ever has enough money to do everything or enough people. So then you have to figure out, well, what would be the things that we could do? Where are the standards needed in type 1 diabetes research, right? If we had those, and to answer your question there, Kendra, right? If we had those, would it be easier for you then to be able to go and look at another study and do the same thing? This is really where, FAIR and these other things come about, but they're not magical, right? They do require the community itself to have some agreement that they would like to do this. Right? It's not really something that can be imposed.
Yeah, it has to sort of like be an organic acceptance. I'm going to do one more poll now. This other poll, I hope we can find it. Yes, there it is. Okay, and so this is this poll I've launched basically is asking, what is the most critical challenge to implementing AI and machine learning in T1D research? And the answers include shifting data patterns, dynamic data collection, scarcity of data and data sets for replication, heterogeneity and correlated data, and data quality issues. I think we tapped on all of those already. But yeah, there's problems. There's some problems. But I mean, I think this little mini consortia, just sort of talking about it and bringing it up can possibly sort of act as a little ember to maybe get, JDRF and others who have more power to start considering, what would that look like? What kind of, maybe dkNET is a place to start in coordination with T1D Knowledge Portal or something like that. But it looks like for this poll, and I'll share the results, it looks like most people feel that it's the heterogeneity of and correlated data, and also the scarcity of data and data sets for replication. And if anyone would like to kind of comment on that, that'd be great.
I think those go hand in hand. There's both a lot of data that's being generated, but if it's heterogeneous and uncorrelated, then it's scarce, right? It's still, right, you don't have enough to do for any particular question. You never have enough.
Yeah.
I think another place that I would love to see sharing too, and I think as we build our mechanistic models, is how do we share our algorithms? How do we, so we can link them and integrate them? Like, because you're working in this silo, and then your study is doing this, and then you see bits and pieces, but to keep up with all the literature, unless you're in COVID, not attending meetings as much, and it's more virtual, so you're picking and choosing. It's hard to see what's being done out there, and having a shared location where, if you're working in a specific area and you want to start building integrative models, how do we pick up those algorithms that'll help us in our machine learning techniques?
Yeah, these are questions that companies like ImmuneAI, a company sort of started from a group who came out of MIT and is now based in Tel Aviv and UCSF. And they're really diving into, and they do have an interest in type 1 diabetes and autoimmunity. And, again, they have a business model, so that's going to drive it. But I think that things can be learned from the way they play in that space too, if they would share some of that. I guess in the last a couple of minutes, I'd like to open it up to questions from the chat. And if anyone wants to sort of in the attendees, I'll look to see if any hands are up. You know, just feel free to ask the panelists any questions, or if anyone would like to offer any other thoughts on the way forward to kind of organize and structure this sort of new way of doing science, right? That incorporates data and the wet lab. Anybody? I don't see any questions. Okay, well, I'll ask my own questions. So, I mean, how do you envision this? I mean, if you think about this, we've got sort of clinical scientists in the room here. We've got MD PhDs, you've got bench scientists going on, you've got data scientists, and you've got, you know, a graduate student who's really deep into sort of machine learning.
Monica, it does look like a question came through.
Oh, okay.
Yeah, I don't want to interrupt your question.
No. I do see it. I mean, how can we I guess circle the wagons and in all these different disciplines? Does anyone have any first step ideas, actionable plans to do it? What would be the first step to do it? And then while you're thinking about that, I'll ask this question from the audience, which is, do the panelists believe that the medical insurance companies may be sitting on large amounts of useful data with respect to T1D? And if so, have any efforts been made to mine that data? I mean, there's certainly Dexcom is getting kind of a lot of actionable data in terms of like lived experience. But I don't know about genetic data. Maybe you guys can comment on that.
I'm not sure this quite answers your question, but you know, there is this thought with the electronic medical health records, right? That we are all our own, everybody is a participant in a potential experiment, right? Everybody who goes to the doctor and has a medical record that is there. And at least the big medical schools and medical centers are really working towards making those data sets available to, you know, they're being very careful about understanding that this is a huge source of information. It should not just be mined by insurance companies, right? But that it should be made available through the proper consenting and other ways of making sure that this data can be shared with the proper security. So I know at the University of California, right, there's a big effort to develop principles around this, right? That if people are coming into the doctors, that this is a potential source of information. We don't want it abused. There's been cases like that where Google and others have gone in and, you know, mined it. Now they have the capacity to take data at this scale. But I know at least UC was very careful about saying we have to do this in a principled way to make sure that this data is used for the benefit of the people who are putting it forward. It's non-exclusionary. There's no exclusive contract with so-and-so who's allowed to come in and look at it. So there are real efforts. This was one of the pain, as I understand, inflicted on medicine by going to electronic health records was really meant for this type of promise, right? You basically now have the data from individuals who are going about their lives. And if the standards come in and other things, it's a potential source of information, but it's very sensitive information and needs to be handled with utmost security and also sensitivity to potential abuses of populations. So it is going on, at least in the big medical school.
Yeah, and T1D Exchange in Boston also has, you know, this kind of approach to try to engage and garner these different clinical data sets from type 1 diabetes patients to be able to kind of create a better picture of their lived experience.
I also know there was some efforts done by a group, I think it was NIH funded, HL7, that did for electric medical record standards. So getting domain experts to help come up with the questions or the type of fields or features or elements we want to capture. So then we can see some consistency across the board. And I don't know how far that went, but I do know that was, you know, in the very early days, they were trying to do that for specific diseases. And that would be very useful.
Yeah. I I will circle back, I guess we have 5 more minutes remaining, just to sort of, kind of think about what would be an actionable item that would be a first step towards creating some kind of, consortia that really sort of unsilos what we're talking about today. I mean, would it be a discussion with JDRF? Would it be a discussion with dkNET? Would it be a discussion with the NIH?
I probably all of them. I mean, in the sense that I'm not a type 1 diabetes researcher. I don't know the state of your community because it is a community, right? The community of researchers who work on type 1 diabetes. Whether there's what is the main challenge to start to think about this as a consortia of groups who are all working towards something that is shared, and that there's certain problems, even as everybody is independent, that need to be, that if they are addressed, right, would make it easier for everybody to do their work. Is there an appetite for that? Because if there is, then it's just a matter of putting the convening structures in place, obviously getting some support. If there isn't, then the problem is how do you inculcate that, right? And start to get researchers to think that way. And in the various things I participated in with you, Monica, sometimes some people are like, no, right? This community doesn't want to do this. Whereas in a group like this, it's quite clear that there's an appetite for this, especially because there's so much informatics here, and informatics depends on data, and people want to get there, want to get data. So I think it's really taking the temperature of what type 1 diabetes researchers want to do, basically, and then putting a convening structure. I happen to think that community organizations that are convening structures are really important. They're very difficult to fund, very difficult to support, but they are the things that sort of cross across all the disciplines and bring them together towards these larger shared goals. And
well, that's one of the missions of the Sugar Science, is to provide a platform for conversation so that we can have a global consortia and people, scientists sort of connecting in this sort of, in this way.
Right, but it's not just conversations, right? There needs to be a mechanism to follow up with that. So I see the difference in disciplines where there is this sort of sense of shared purpose. There's cross project working groups, right? There's other things that come into existence because they say, hey, we really need to get these things going. A lot of those are funded by NIH, right? They said, okay, we can get all these people to work together, and then there's going to be this coordinating center, and that means that you're all going to have to coordinate in some way. You're all going to have to come together over this. So I think there's many mechanisms for it. And certainly dkNET, right, would be more than happy to sort of help figure out how that goes. Because I think what you asked for, Kendra, shared algorithm spaces for that. There are platforms for sharing those types of algorithms. Now, none of these are entirely simple, right? But there are means to do this. If this would be something interesting to people, you can set up platforms.
I think too, with in terms of getting more sharing, is being inclusive. Like when we write our grants, we include people from these data sets that we want to use so we can have that domain knowledge. And we also have the collaboratory nature and not try to just use a data set and keep it in their silo. That kind of that will start opening the door, I believe. But I mean, that's just my thoughts on that.
Yeah, when we look at the papers that came out of, the Sepica lab there, or the Sepica grouping, right? They were pulling in many different researchers. And I think that kind of a paper can be so informative because it has all these different, it's like a hive mind, right? And they are all adding value when they write this paper. And with the sort of the TCR approach is very popular. So it's, I think that's one way to do it. Everyone's going to be on this paper, and it's going to be a Nature paper, and that's exciting. But here's one more question. We have like 1 more minute, but this is kind of a curious question, because why nowadays most research is on type 1 diabetes instead of type 2 diabetes? A computer science researcher very new to diabetes. Well, I would have to say, from my perspective, I think T2 diabetes is gaining a lot more research just because of the market size and all the forces that kind of drive that, like the market, the size of the type 2 market, the size of the type 2 population is driving research in that arena. And it is in some ways, like a threat to type 1 diabetes research because it's a smaller group. So yeah, computer science researcher, come join type 1 diabetes research, sure.
They're fundamentally different diseases. They have the same manifestation, but the mechanisms are fundamentally different.
Yes.
So they have this diabetes as an outcome, but what the principles are very different.
Yeah, totally. But Monica, I was also going to say it's a credit to sugar science and efforts like that question could even be asked, because that suggests that the visibility of what's going on in type 1 is growing, which is really exciting.
Well, I hope so. I mean, personally, I have skin in the game, as we say. Most of my volunteer, team on the sugar science also have close connection to type 1 diabetes. We all want to see the symptoms mitigated and ultimately cured. I mean, that would be just a dream. So we appreciate everyone's contribution here to this conversation and this sort of like participating in this new way of talking about it. And maybe it's a chance when it's we post it on our YouTube channel, and we do get a lot of sort of different back and forth comments there, and it's another chance for people to hear it and think about it, start thinking about ways we can push it forward. And thank you all again very much for taking the time from your day and your fantastic work that you're doing. I so appreciate it, and I hope you have a great rest of the day.